Long, good evening. How are you? How are you, Shillong? Good? Yes. How was the school today? Yes, children. How was your school? How are you? Okay, just give me a hi if this is 11th Shillong, Biology. Yes, just give me a hi. Just wave your hands. Yes. 11th Shillong, Biology. Is it? Or I am in a wrong class? I am watching a wrong class. Is this my class? Yes, children. Okay. Uh, yes. Just wave your hands. Okay. Fine. So, how was your school today? How was your school? Tiring? Seems to be. Is it? Good? Okay. So, what were we discussing? Discussing biomolecules, isn't it? The last class, we're talking about biomolecules. I'll just write down the names of the things we completed. We completed amino acids. We completed proteins. We completed lipids. We completed carbohydrates. Just let me know if not. Okay, we completed nucleotides, or we also completed nucleic acids. Is it? Did we complete carbohydrates? Let me know, please. I remember amino acids, proteins, lipids, nucleotides and nucleic acid we completed. Did we complete carbohydrates? Tell me, children. Yes. Did we complete carbohydrates? Yes, no? Yes, Shillong. Please tell me. Tell me, please. Did we complete carbohydrates or polysaccharides? Okay. We did not do polysaccharides. Have we done polysaccharides? Okay, let's start up with polysaccharides then in that case. Okay, fine. Got it? No issues. We'll proceed with polysaccharides. So, poly means many, saccharides means sugar units. So, what happens is basically, if there is 1 unit of sugar, it is said to be monosaccharide. We know many monosaccharides. If, for example, you talk about glucose, digestion, remember we talked about glucose, we talked about fructose, we talked about ribose, deoxyribose. So these are the typical examples of monosaccharides. We call these molecules to be what? We call these molecules to be monosaccharides. These are monosaccharides. So these are monosaccharides, and these monosaccharides, if are attached to each other, or we can say they are 2 units, we say them to be disaccharides. For example, you have is maltose, sucrose, lactose, isn't it? Remember the digestion chapter. So maltose can be broken down into 2 subunits of glucose plus glucose. Sucrose is similarly broken down into 2 subunits of glucose plus fructose. And lactose is broken down into galactose plus glucose. So these are what? These are the disaccharides which are typically important when you talk about sugars, when you talk about the units of sugars. So these are the examples. You to understand? Okay. So these are, if a single unit is present, is said to be monosaccharide. If 2 units, it's called as disaccharide. And similarly, if you have many sugar units attached to each other, we say them to be what? We say them to be polysaccharides. So if it is many units, it's called as what? It's called as obviously poly means what? Poly means many. Example you have is cellulose, starch, glycogen, inulin, chitin. So these are what? These are the particular examples of the polysaccharides. Like maltose, sucrose, lactose. You have glucose, fructose, ribose, deoxyribose are the examples of monosaccharides. So in this way, we have poly and monosaccharide, di and monosaccharides as the typical units under the carbohydrates. Clear? So what was the section in which we got this? One question for you. Yes. Answer this question quickly, Shlong. We did this in first class itself. Answer please. Yes, children. What do you think should be the answer? Shillong is very tired, I guess. Is it? You people are very tired. Are you? Seems so. Yes, you are. Yes, children. Quick. Quick, quick. It's very easy. This we did in the first class. You can just flip through the notes. You'll find it in the first class biomolecules. We have done that. You can cheat. No issues. Till you have not prepared for test, you can flip through the pages. Yes, quick. First class biomolecules. See? Name the pool it was present in. Quickly. Yes, children. No, yes, tell me. Remember, we separated, first of all, we crushed it in TCA, the tissue, and then we filtered it out. We got two phases. One was retinate, other was filtrate. The filtrate was that, I gave you an example also, that you prepare tea at home. The tea would be what? The tea would be the filtrate part, and the tea leaves that will remain in the particular whatever kind of thing you are using it, sieve to filter it. The tea leaves will be retinate. So you have two sections, the filtrate section, acid soluble. The retinate section, acid insoluble. Acid insoluble section will contain all the polymers. Acid soluble section will contain all the monomers. So the polysaccharides are what? Polysaccharides are polymer. They are having a molecular weight of more than 10,000 Dalton. Remember, the molecular weight would be what? It would be more than 10,000 Dalton. So with this molecular weight, they will be appearing in the acid insoluble part, or also called as retinate section. So this would be the correct answer. Clear, children? Am I clear? Yes. Yes, exactly. I guess there is some net issue also. So it will be found in which section? In the acid insoluble or in the retinate section. Is it clear, everybody? Just give me a thumbs up if it's clear. Yes, children. Give me a thumbs up. It must be clear that day itself, but I guess you did not revise it. I have sent you an assignment of chapter excretion today. Please make sure you're going to do those questions for the next week. That is whole week time given to you for just one chapter. Please make sure you are doing them. Okay? Fine. So let's resume. Come back. Then what we have is, we'll talk about polysaccharides. So polysaccharides are what? Let's define it. They are long chain of sugars. They're long chains of sugars and made up of many monosaccharide units. Let's see how the monosaccharide come and form a bond together. So this is one of the glucose molecule. I'll just start off with glucose and not make the matter more complex. We'll just finish up with glucose itself. This is one glucose molecule. This is another glucose molecule. What do you have? Let's remove, as we did in other molecules, we'll do the same for polysaccharides as well. So we are going to remove this water, see, H2O removed, and what you get is, this is one glucose, okay, this is another glucose, and what you get is finally so this is what is basically glucose-glucose bonding. So this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon and CH2OH, sixth carbon. First, second, third, fourth, fifth, CH2OH, sixth carbon. So at the particular place we can notice over here what the changes are going to take place. That is, this is the first carbon, this is the fourth carbon. So there is a bonding between the first carbon of the first glucose molecule with the fourth carbon of the second glucose molecule. So there is a removal of water, and this particular bond that you have seen here, this particular bond is said to be what? This whole bond is said to be glycosidic bond. This is quite important. So the bond formation will take place in such a way that you need to remove, like all the dehydration condensation reactions we have studied till now. Similarly, you have to remove with the same pace, you have to remove one water molecule, and with the help of the water molecule, removal of the water molecule, what you get is COC bond. See, C over here and O in between. So this bond is said to be what? This bond is said to be the glycosidic bond, and this glycosidic bond is present where? It is present in between, this is B/W, between monosaccharide units. So if it was peptide for 2 amino acids, if it was phosphodiester for 2 nucleotides, if it was ester for lipids, it is glycosidic for polysaccharides. This is the glycosidic bond formation taking place. Clear? Are we clear, everybody? Okay, very good. Nice. Keep it up. Let's do one small question. Let's see how much we remember. Do this question. Match it. Quick. Everybody has to do this. Not only the person on the keyboard. Everybody, all of you have to do this, please. On your notebook, match the particular with their bonds. Yes, children. Match it. Everybody, all of you. I can ask anybody among you to write on the keyboard. So all of you have to solve this. Done? Solved? Give me a thumbs up if you have done it. Yes, children, please. Give me a thumbs up if you have done this. Yes. I cannot see a single thumbs up. Please. Almost done? Okay, do it. Just take 1 more minute. Complete it. You're flipping to the pages? Oh, okay, you can do it for today. No issues. Done. Quick. Done, everybody. Enough. Done, children. Yes, no. How much time you are taking just to match this? This should have been done. Yes. So I'm going for a short nap. Just let me know when you are done with this question. I'm going on for a short nap. Power nap. Shall I? Yes, answer please. Good. Very good. Yes. Nice. Good. Very good. And last one is obviously whichever is left. Okay, very good. Keep it up. Everybody, clap for yourself. Nice one. So lipids, they are having ester bond. Proteins, they have, among them is peptide bond. Polysaccharides, just now we did it, glycosidic, and nucleic acids, they have phosphodiester bond. Very good. Keep it up. All of you. So let's go back to polysaccharides. So these are the bonds present between different biomolecules. Let's go back. So this was the formation of glycosidic bond by removal of a water molecule. And then let's see what are the different types of polysaccharides that we are having in nature, that we are having in abundance around ourselves. So first of all, if we talk about polysaccharides, we have certain polysaccharides which are said to be starch. We have all studied starch. What is starch? Starch is the storage form of carbohydrate in plants, isn't it? Then we have it's a polymer of what? It's a polymer of glucose. It has a very special helical structure. Or helical chain, which can trap iodine to give a blue color. So you must have done this in school also. The teacher must have shown you potato starch test, where she must have cut, or teacher must have cut it in two sections, potato, and then stained with starch, and then shown you that you see this is blue color. So wherever you see blue color, that is what is with iodine, obviously. That is basically a sign that starch is present in that particular molecule, in that particular whatever the content is. So starch is one of the polymers of glucose, one of the polymers I am telling you. But because we have another polymer also, that is glycogen. So this glycogen is often said to be animal starch. Obviously animal starch means it is stored form of carbohydrate in animals. So animals also store carbohydrate, but in a different form called as glycogen. So starch is to plants and glycogen is to animals. So it's said to be animal starch. Now glycogen has a typical straight chain structure, or we can say straight chain polymer of glucose. It is also a polymer of glucose, but if you try and have a starch test, it does not give a blue color. With iodine. This is typical thing present in glycogen. They do not give a blue color with iodine. Third carbohydrate, cellulose. This is structural carbohydrate. Present in cell wall of plants. This again is a polymer of glucose. And this is found in paper plus cotton fibers. And after this we have is inulin. This is a polymer of fructose. So up till now A to D, all the examples from A to D are homopolymers. That is made up of single type of monomer. Apart from this, I would add one more thing, that is chitin. So just edit it A to D to A to E. Chitin. This is also a structural polymer. This polymer fructose is a storage polymer. This is again a structural polymer. And this structural polymer is basically present where, if your question comes to you, this is found in the outer covering of, or is exoskeleton, that is the outermost covering. Like you must have seen an insect, it must be very tough from outside. So the toughness is basically because of the exoskeleton, and this is made up of chitin. And sir must have told you that in fungi also, the exoskeleton or the cell wall is made up of chitin. So these are the most important two places where you can find chitin, that is the exoskeleton of insects and cell wall of fungi. Now there is one more thing, is reducing and non-reducing sugar. What is reducing? Now if a sugar is able to convert Cu2 plus to Cu plus in Benedict, in Felling's solution, we call those sugars to be reducing sugars. Now the right side of any sugar, that is polysaccharide, the right side, let's say right side of the glycogen. Okay, so the right side of the glycogen, or let's say right end of the glycogen, is reducing. That means it will reduce Cu2 plus to Cu plus, and left end is non-reducing. It will not convert the Cu2 plus to Cu plus. So this happens where? This happens only and only in which solution? In Benedict and Felling solution. Clear? Are we clear up to this, everybody? Sorted? Am I clear, everybody? Okay, very good. So this is what is we call as the typical sugars. all these examples are of homopolymers where we have only single type of monosaccharide or monomer forming multiple units by glycosidic bond and leading to formation of a polysaccharide. So these all are what? These all are homopolymers. So this is all about your carbohydrates. Let's start another topic, that is primary and secondary metabolites. You don't have any doubts in this? Everybody just give me a thumbs up, please. Yes, if the things are clear, obviously. Things are clear? Topic is clear? Yes. Thumbs up, please. Yes, children. Get up. Yes. Is there a net issue? Or I am not able to see my class? Yes. Are you giving me a thumbs up? Or I am not able to see you, I guess? You are, I guess. Okay. Yes, yes, yes. Very good. All of you, the boy on the first bench. Yes, child. Things are clear? The boy on the first bench. Yes, child. Very good. Okay. So we'll start a new topic, that is primary and secondary metabolites. So maximum time, since this question is asked for a difference between, so I will deal in the same manner. We'll write out some different differences between primary metabolites and secondary metabolites. So what are primary metabolites? Anybody? There are few who have completed this chapter in school. What are primary metabolites? They are sources of energy. They are not source of energy. Manufactured by green photosynthetic plants. Manufactured by plants, fungi, microbes like bacteria, so we have to depend on plants for these energy sources, but in this case these products are not of energy value, but yes, they have value in human welfare. So you talk about human welfare, they are because of the secondary metabolites. Let's see some examples. We have is amino acids, proteins, lipids, energy sources like sugars, etc. For all these things, we need to have what? We need to have the plant resources. So when we eat plants, then only we get these resources. So the examples over here are flavonoids, rubber. So rubber is not important energetically, but yes, rubber is of great human welfare usage. We have essential oils, we have antibiotics, we have colored pigments, we have scents, gums, spices. So all these things are what? All these things are secondary metabolites. So secondary metabolites are not going to help you out in energy purposes, but yes, they are very, very important when you talked about the other usage, that is of human welfare. So they are thought to be of what? They are thought to be of ecological importance. They are important ecologically. Am I clear, everybody? All of you, yes. So if you need to have carbohydrates, that would be what? Primary or secondary metabolite? If you are eating carbohydrates, what is that? Primary metabolite or secondary metabolite? Tell me. Are they primary or secondary metabolites? Carbohydrates. Copied, everybody? Have you copied? Yes, they are primary metabolites. Why? Because they are sources of energy. We have copied? Proceeding. Shall I proceed? Next heading. So you must have studied about catalyst, isn't it? So what are enzymes? Enzymes are the biocatalyst which increase the rate of the reaction without getting consumed themselves. So all enzymes are protein. But however, we know there are exceptions in biology. So we have two exceptions. One is ribozyme and two is ribonuclease P. Ribozyme and ribonuclease P are two enzymes which are not protein, but both of them are what? Both of them are RNA in nature. That is nucleic acid. So we have studied the different structures of proteins, isn't it? So I have a question for you. Enzymes have blank structure, obviously structures. It can be one or more than one as well. First option is primary, secondary, tertiary, quaternary, both. Fifth option is both 2 and 3. Sixth option I'm going to give you today. And sixth option is both 3 and 4. Yes. Tell me. Tertiary. Now what if I say yes, tertiary structure is correct, but what if I say that they have both secondary as well as tertiary structure? Some of the enzymes, maximum enzymes, they have tertiary structure. This is very much true. I have taught you this also. Very good. But however, we have some examples of the enzymes where we talk about the secondary structure of the protein as well. So enzymes, they are having both secondary and tertiary structures of protein, both secondary and tertiary structure, that is alpha helix, beta sheet, as well as the particular hollow wool ball like structure formed by different kind of bonds, isn't it? So what we have is enzymes have pockets in their structures, and the best suited structure for this is tertiary structure. So tertiary structure of the protein is the best suited where an enzyme can have a pocket-like structure. This pocket is nothing but this is called as the active site. So if a definition of active site is what? A site which binds to substrate. What is substrate? Substrate is the raw material. And it converts it into product. So if you talk about a substrate and active, quickly let's see what is basically happening. So this is an active site of the enzyme, this V-shaped structure, and a similar kind of structure is present with the substrate also. This is a substrate. So this is S, this is E. So what is going to happen? They are going to bind together, obviously, in this way. They are going to bind, and yes, they will be forming a complete structure. This is what is called as enzyme substrate complex, and this enzyme substrate complex turns the substrate after a while into product. Let's change the color. This pink one is the product. This pink is the product. So it's said to be what? It's said to be enzyme product complex. And finally, this enzyme product complex dissociates from each other. So we talked about this thing earlier also, that enzyme never undergoes changes. Okay, this is waste. I don't know why this is happening. This is E plus B. Now this E is again ready for the next round of reactions. It can again bind to a new substrate molecule. So this is what is basically happening. So what is an active site? An active site is a site which is basically important for what? This is basically important for binding to the substrate. So if you want to bind to substrate, you need to have an active site, and as soon as the substrate binds to active site, it gets converted into a product. Clear? Are we clear with active site thought process? Concept of active site is clear to everybody? Yes. Tell me, children. Is it clear? Yes, children. Okay, very good. So we'll proceed with the next thing. Now, when you study chemistry, you must be having certain catalysts that increase the rate of the reaction, isn't it? We have thought process. We have already an idea about different kinds of complexes that are said to be catalysts. So there are two kinds of catalysts. If you talk about the one which we call as inorganic catalyst, and the other is the enzyme or the biocatalyst. So maximum of the inorganic catalysts, they can withstand or can tolerate a high temperature. But enzymes are proteins, isn't it? We have just not seen that. And because they are proteins, they cannot tolerate a very high temperature. Like if you increase the temperature about 40 degrees Celsius or more, the enzymes, they tend to get denatured. they lose their properties at high temperature. Tend to lose their properties. But yes, we have certain exceptions again. We have certain microorganisms. Micro means small, organisms means living things. Living in thermal springs. So these microorganisms which are living in thermal springs, so I must have shown you what is a thermal spring. You know it is a thermophilic bacteria. Can tolerate a temperature of up to 80 to 90 degrees Celsius. So this again is an exception where we actually were talking about that yes, the enzymes get denatured if you are taking it above 40 degrees Celsius, but at the same time you have certain exceptions about the microorganisms living in hot thermal springs with a temperature of 80-90 degrees Celsius, and yes, their enzymes are active at this temperature as well. So this is very unique thing. This is very a kind of thing which is very important in its own ways. We will study the importance in the next year. So you have to wait for whole 1 year to study its importance, that how we can use these particular stability of the temperature property in our own welfare. Copied? So if you talk about a chemical reaction, isn't it? If you talk about a normal chemical reaction, we have certain points under it. Like first thing we have is rate of reaction. So rate of reaction is what? Rate of reaction is delta P by delta T. Delta P by delta T, what does it mean? It actually means that rate of product formation in a given unit time. So if a chemical reaction has a higher rate, this clearly indicates that the product formation is more in that particular time. Obviously, delta you know is change in time, initial and final timing. So increase in rate of reaction, product formation in that given time period is said to be what? Is said to be chemical reaction. Clear? All of you? Yes, children? Clear? Sorry. Are we clear, children? Today you are so tired. You have to do homework also, excretory waste and its removal. I have given that on your groups. Seem to be very tired today, is it? Okay. Have some rest, go back home, take some rest, have good nutritious food, and then you can start solving the questions, assignment. Okay. So we have certain factors, let's see. Now, the factors which affect the rate of reaction, the first and foremost thing, affected by temperature. How? Now, if you increase the temperature by 10 degrees Celsius, the rate of reaction doubles. So if you want to increase the rate of reaction, obviously you need to increase the temperature by 10 degrees Celsius. Obviously, you should not go up to 40, otherwise the enzyme will get denatured and your rate of reaction will not proceed because enzyme will be inactive for the rest process. Now, similarly, if you decrease the rate by 10 degrees Celsius, rate of reaction is just half. So an increase or decrease in temperature is going to affect the rate of reaction in either in a twofold way or just decreasing it by half. Let's see what is the rate of reaction when you carry out a process with enzyme and without enzyme. So we'll talk about a reaction we have studied in breathing exchange of gases. That was a reaction where the CO2 diffused in the RBC, it combined with water, and then there was a reversible reaction catalyzed by an enzyme called as carbonic anhydrase. Remember? And you get is what? H2CO3. Now let's see this reaction in presence of carbonic anhydrase and in absence of carbonic anhydrase. Now when you have this enzyme, in presence of enzyme, let's see first. This is the fastest enzyme known. Enzyme known. So in the presence of this enzyme, the rate is somewhat 6 lakh molecules in a single second. See, such a huge rate. And when this enzyme is absent, the rate is only 200 molecules, and that too in a single minute. So this is very much clear that the rate of reaction or we could say the above reaction is 10 million times more isn't it? So when you apply carbonic anhydrase, the rate is somewhat how much? 10 million times more. And if you apply only without the enzyme, this reaction will produce only 200 molecules of H2CO3, that is carbonic acid, in a single second. So it's a huge difference. There's a huge parameter lagging behind in between, which is able to basically manage through the things. So in this way, we can actually have an idea that yes, how the enzymes are important, how they are. Just let me know if this color is comfortable to your eyes. So if it is not, just let me know. So this is the particular thing which is making the whole process of enzyme catalyzed reactions so important. That's why we always prefer an enzyme to catalyze the reaction in a way that we can have the particular product formation at a very, very huge rate. So I would always like that 6 lakh molecules of carbonic acid is formed, and that changes to H plus HCO3 minus, and my transport of CO2 is executed, isn't it? So this would be always preferred by my body, rather than having it in an uncatalyzed way, producing only 200 molecules in a single minute. So this actually makes a huge difference, isn't it? What do you think, people? Yes. What would you prefer? With enzyme or without enzyme? What do you prefer? With enzyme or without enzyme? No idea. Yes. Yes, Shillong. Catalyzed or uncatalyzed? What is taking so long? Are we connected? You are, I guess. I can see you. Yes, obviously. Okay, fine. So let's see a graph where we can actually depict the two values. We'll try and make a graph. Can you see the difference? So the purple graph, this particular dark colored, this shows the amount of energy you need to cross the transition state. Now what is transition state? Just wait for a single minute. We'll see the transition state over here. See, There are there are four stages. This is the initial stage. These two are temporary or transient stage, or you can say the transition stage. And this is what? The final state. So you have different stages, you have different states where you can actually talk about different kinds of processes, transitions taking place. So in between the initial and final, these are energetically stable products and the substrate. These are energetically stable states. But the two states in between, that is ES complex and EP complex, that is not stable energetically. That needs a particular amount of energy. Every molecule, they need a particular amount of energy to cross this transition state, and this is what is we are depicting in this graph. So this varies from each molecule, varies from molecule to molecule, that how much transition state will require, how much energy will the transition state will require to cross this substrate barrier, energy barrier, and convert to product. This is product P. So the substrate formation, the substrate and the product formation from the substrate needs to cross an energy barrier. This is the particular energy barrier. So what does an enzyme catalyzed reaction does? This is an enzyme catalyzed reaction. I'll just highlight it. This is this pink one is an enzyme catalyzed reaction. The pink graph. This thing, where we actually need a lesser amount of energy to cross that energy barrier. But in a particular reaction which is not catalyzed by enzyme, you need a huge amount of energy to cross that particular energy barrier. That is the significance of transition state. So an enzyme, what does it do? Number one, how does it act? Have you copied this graph, everybody? Have you copied this graph? Yes, children. Tell me. Okay, very good. So what is the overall thing going on over here? S needs to get converted into product. But this is basically what? This requires an energy. Let's talk about this. Requires activation energy to cross the energy barrier. Isn't it? So it requires what? It requires an activation energy to cross the energy barrier. Now this activation energy is lowered in presence of an enzyme. So if you put an enzyme in a reaction, what consequence you are going to have? You are going to have a lowered energy, isn't it? Lowered activation energy. So this activation energy is lowered in presence of an enzyme. Clear? Isn't it? So not only activation energy, but this also lowers the time of the reaction. Like just now we saw, it was 6 lakh molecules in a single second and only 200 molecules in 1 minute. So depending on that, you have what? It was minute or it was hour? I guess it was hour. Sorry, this must be hour. Just rectify this. This is hour, not minute. It's more complex than you were thinking. Just rectify this, please. It is 200 molecules in a single hour, whereas you have a 6 lakh molecules in a single second. So this does what? This actually lowers 2 things. Number 1, the time. Number 2, the activation energy. So enzyme is actually doing a 2-fold increase in the reaction rate, or you can say acts in a 2-way to increase the reaction rate. Number 1, it lowers the activation energy. Number 2, it decreases the time for the whole reaction. But let's assume a situation where you have to go to a building having 20 floors. Okay, you have a floor. On the 20th floor of the building, you have a flat. Now you are supposedly going to that particular flat. You have 2 options to go to 20th floor. The first option is either you can use a lift or you can use the staircase. What would you use? Yes. What are you going to use? A lift or a staircase? Yes, why? Exactly, I will do the same. Why? Why lift? Why not staircase? Just tell me please. Why lift? Why not staircase? Exactly. Yes. If you go by stairs to 20th floor, finish. You will not be left with any energy to do anything else. You will just go home and sleep. No study, nothing. You will not be left with any amount of energy. Yes. Plus, what? Who is going to reach first? If you go by staircase, I am going by lift. Who is going to reach first on 20th floor? Yes. Come on. Who is going to reach first? You are going by staircase, I am going by lift. Who is going to reach first? And fresh, obviously. The question is clear? Obviously, why? Because lift provides a surface. Lift is providing a surface where you can just go and reach there, and once you are there, you will be saving your time as well as energy. Similarly, your enzyme is also providing a surface. Your enzyme also provides a surface, the same surface said to be as active site, where what is being done? You are saving the time in conversion of substrate to product, and you are saving the energy for the same purpose. So what is enzyme doing? Let's see. What is enzyme doing? How is enzyme acting? So enzyme provides a surface. And what is the surface? Active site, where breaking of old bonds of substrate and making of new bonds in product takes place. So enzyme's active area or active site is important in this way. Clear? So this is the whole concept of how enzymes lowers activation energy. So if you get to have this question, but how enzymes lower the activation energy, you have to write only this much, that enzyme provides a surface or active site where breaking of the old bonds of the substrate takes place and making of new bonds in the products take place. So the end result is what? So the conversion of S to P takes less time plus energy. Which energy? Obviously activation energy. And this activation energy was used for what? For crossing the energy barrier, isn't it? This activation energy was needed for what? What purpose? This was required for crossing what? Energy barrier. This thing. This is the energy barrier. This is the energy barrier. So you need what? You need an enzyme to provide that surface where you can break first of all the old bonds, make new bonds and cross the energy barrier that is present in the transition state. So this energy barrier is said to be activation energy, which is basically lowered by an enzyme. Yes, clear how enzymes act? Are we clear? Yes, Shillong. Are we clear, children? Okay, very good. Keep it up. Any doubts up till now? Any doubts? Any doubts on enzymes? No doubts? Sure? Okay, let me ask a question. You saw this particular reaction, isn't it? We know what is exothermic and what is endothermic. So what is this? Is this an endothermic reaction or exothermic reaction? You know exothermic? What is exothermic? If you put limestone in water, it gives a huge amount of heat. That is an exothermic reaction. That is, the reaction is going to liberate heat. And what is endothermic? If you are heating up the reactants or the substrate in the initial phase of the reaction, that is endothermic reaction. So endothermic reaction is going to consume energy. Exo is going to release energy. So what kind of reaction is this? Is it exothermic or endothermic? Tell me. In either of the cases, S has to cross the P barrier. S has to convert to P by crossing the activation energy. So what is this? Exo or endo? Yes. See, what happens in an exothermic reaction, S is higher than P. See, if substrate is at a high energy, product is at its lower energy. We are having what? Potential energy over here. So product is at, if it at lower energy, it is going to release heat, isn't it? Substrate is higher, product is lower, it will be releasing heat. So this is an example of exothermic reaction. If the substrate would have been lower and product would have been higher, it would have been an endothermic reaction, the opposite case. Clear? So this is an example of an exothermic reaction. Why? Because substrate is higher than the product. Am I clear? All of you? Am I clear, children? Okay. I guess I am. Okay, very good. So this is like, we will wrap up over here. We will start from the next, in the next class, we'll talk about the factors affecting rate of reactions. Just remind me about this. The factors affecting we'll start up from this in the next class. Fine? Okay. So you can, no doubts, nothing, no confusion. Okay, just go back home and revise everything, whatever has been taught today, and try to do assignment from the extrusion part that I have WhatsApp sent on the WhatsApp group today only. Just try to do those questions and let me know in the next class. Yes, we will be starting locomotion and movement in the next class, because since we have just small topic left, factors affecting and then cofactors and enzyme classification, that will be done in like half an hour. So what's the new chapter we are going to start? Locomotion and movement? Shall we start with that? Tell me, 20th chapter of your syllabus. Can we start with that? Yes. We can, isn't it? No upcoming tests? Nothing? Nobody's going to have animal kingdom? Yes. Tell me, what to start next? Okay. So shall I start with locomotion and movement? I will start with the same chapter, locomotion and movement, in the next class. Okay, fine. Okay, thank you so much. We'll see you again on Monday. Please revise the chapters, go back home and study. Okay, tata, bye-bye. Take care. Bye.